right, today for this segment, I would like to draw out the 5 steps of mitosis. Remember, mitosis is a kind of cell division. It's where we take one cell with a diploid number of chromosomes, and we split that cell into 2 identical copies of that original cell. And so I want to walk you through the 5 phases. We're going to start with the diploid cell with 4 chromosomes in it. That's our diploid number that we're starting with. So hopefully by the end we end up with 2 diploid cells with 4 chromosomes each in it. And up here I have written out for each of the steps of mitosis, I'd like you to know 1 to 3 significant things that happen in that particular step. So let me draw this up. Here's our cell boundary, our plasma membrane on the outside. And don't forget that we have like a nucleus that is intact for this step. So we have a nucleus. I'm just going to write that up really small here. The nucleus has not like sort of disappeared. And we have also our diploid number. So in like kind of before we begin our cell division, before we begin, we're going to have 4 chromosomes inside of our nucleus. But at the end of S phase, right, we will have copied all of those chromosomes. So we actually have added, after S phase, we've added another set, right? Identical copies. That's what happens in DNA replication. So before mitosis, all of that has sort of occurred inside of the cell. Now, DNA packs up, and so one of the things that has to happen is your DNA has to organize itself, and it has to condense into these like very thick cords. This allows for your cells to sort of organize and then pull apart these chromosomes in an even way so that you don't get too many or too few, or you don't lose like a gene or something like that. So packing up finishes, it starts in G2, but it finishes at the top of mitosis and prophase. And then all of our sister chromatids, so these are these identical copies we have made in DNA replication. They find each other and they actually link with a little sticky centromere, this protein in the center that holds those sisters, those identical copies together. The last thing that happens is our centrioles. If you're an animal cell, we need centrioles to kind of anchor off of. And so the centrioles are kind of a star-shaped organelle. And what they're going to do is they have divided and made like sort of a copy in G2, and those will start to migrate towards the poles. So that is the end of prophase. The second step of mitosis is prometaphase. And in prometaphase, one of the first things that happens is the nuclear membrane disappears. And so I'm going to draw this as a dashed line to sort of indicate on this diagram that we just had the nuclear membrane, but now it has like sort of disappeared. The other thing that is significant in this particular step is the centrioles, which started out in prophase. They are migrating to the poles. They reach the poles. They are these star-shaped sort of organelles, and they reach the poles, and then they actually grow out their tubes to sort of connect to each of these like sisters. And so one tube is going to grow out and connect to one side, and then the other side will also connect to each of these like sisters. This sets up what we call the mitotic spindle. This spindle then can like sort of move and move these chromosomes back and forth until they're in position so that we can divide these chromosomes equally. So that's what happens in prometaphase. The third step of mitosis is metaphase. In metaphase, because we've just connected and linked the mitotic spindle to these sister chromatids, all of the sisters are pushed back and forth until they arrive and line up as if they're on an imaginary plate in the center of our cell. And so here are our sisters. They're still linked at the centromere, so we still have that centromere dot in there. And our centromeres are still, centrioles are still like sort of locked onto those sisters. So they have like sort of pushed and pulled these sisters until they line up in the middle of the cell equally. Now we do something called the M checkpoint, and the M checkpoint is an opportunity for enzymes to run this plate. So they run sort of this metaphase plate, and these enzymes check to make sure these sisters are equally connected, so that you have one sister connected and will move one direction and the other sister will move the other way. And only then can your cell evenly divide these chromosomes. So those are the main points that happen in metaphase. The fourth step of mitosis is anaphase, and there's really only one thing that happens in this step. Our sister chromatids split apart. So remember, our sister chromatids were just on this metaphase plate. They have just like sort of lined up like so, and they're linked at first with this like centromere. But what's going to happen to these particular chromosomes and how I would draw this is I would draw them where they're just slightly apart, like so. Remember, we started with a diploid number of 4. We have our centrioles that have locked on to each side of those sisters. So we have those up here as well. And then what happens is that centromere, that like sort of sticky area in the middle, gets like kind of broken down, and then the sisters separate. And the sisters then, once they separate, will be pulled to either end of the cell. So as soon as they're broken apart, then they pull equal amounts of sister chromatids. Once those chromosomes are split, equal amounts of chromosomes will migrate to the opposite ends of the cell. And that's what happens in anaphase. The last step of mitosis is telophase and cytokinesis. These happen almost simultaneously, although 2 different things are happening. And so I want to walk you through both of them, but note that they're happening at the same time, so they're often given as a set. In telophase, what's happened is all of your chromosomes will migrate to the end of the cell. So remember, we have a diploid number of 4. We had 8 chromosome potentials in our cell at the top of this mitosis. And so now we have 4 that have migrated, 4 chromosomes that have migrated to one end, and 4 chromosomes that have migrated to the other end. So in telophase, what's really happening is your nuclear membrane is reforming around those particular chromosomes. So we have our nuclear membrane reforming so that we have a true nucleus again. In cytokinesis, then the rest of the cell contents, all the cytosol, the liquid, like think about like all your organelles like mitochondria and Golgi and ER, will all separate equally so that you have equal cell contents going one direction and the other. And that is cytokinesis, where the cell essentially pinches off and divides the cells into 2 equal compartments, right, with equal amounts of organelles and liquid. So we started our mitosis with a diploid number of 4. We have ended with 2 identical diploid daughter cells. Notice they each have 4 chromosomes inside of them. And this is the ultimate goal of mitosis, is to start with a cell and then produce like 2 identical copies of that cell.